Now there are three basic approaches in 3D electron microscopy. There's tomography, which is what you do when you have a single unique object that you want to image. If you can crystallize them, you do 2D crystallography. But if you can't crystallize them or you don't care to try, you can proceed with single particle analysis. And here the idea is that you would have a large number of copies of the object of interest, and they would be in solution, tumbling randomly. Then you spread them into a very thin film across an EM grid and plunge freeze it. And in the moment you plunge freeze it, all of those particles could be tumbling in any particular orientation, and they're frozen where they are. And so here when we look at an image of such a sample, this is hemocyanin again, and it's a barrel-shaped protein, and so sometimes you see it from the top view where you see the circle of the top of the barrel. In other cases you see a side view where you can see the extent of the barrel. And each particle presents a different orientation of the particle. Then the challenge of single particle analysis is to take each of these individual images and find the orientation and the alignment parameters that are required to align each of these particle images with all of the rest, and then you average them together to produce a much higher signal-to-noise ratio reconstruction. The averaging both gives you all the views put together in 3D, and it allows you to boost the signal-to-noise ratio and try to interpret high-resolution details. And so this is single particle analysis. Now it also exploits the projection theorem, which is again that if you have an object in 3D, like a protein complex, and you record a projection image through it, you now have a two-dimensional projection image of that particle. And if you calculate the two-dimensional Fourier transform of that image, you get a two-dimensional Fourier transform full of amplitudes and phases. And then you can ask the question, what is the relationship between these amplitudes and phases and the amplitudes and phases of the three-dimensional Fourier transform of the original object? Okay, and the answer is that these amplitudes and phases in the transform of a projection image are the same amplitudes and phases that exist on a central slice through the three-dimensional Fourier transform of the object. Therefore, if we record projection images of an object of interest from lots of different directions, and we calculate all of their transforms, we can use them to estimate the amplitudes and phases throughout reciprocal space, because each one will cut through in a different direction. Then we can interpolate the results onto a regular lattice and calculate an inverse 3D Fourier transform to generate a reconstruction of our original particle. Now to illustrate this for single particle analysis, I'm going to show a bunch of slides that come from a tutorial that Steve Lutke made for his software eMan. So this comes from the eMan tutorial. And Steve illustrated that if you have an image of a particle from a particular direction, you can then calculate its Fourier transform. And if it's the first image that you're considering, you can then go ahead and put the amplitudes and phases that you observe on any particular plane in reciprocal space. So here is a representation of reciprocal space. We've labeled the axes spatial frequency in the x direction, spatial frequency in the y direction, and spatial frequency in the z direction. So our very first image of our particle, we just calculate its transform and place it in any plane we like. Maybe the easiest one is to just say that's the xy plane in reciprocal space. Then the next image of our particle, so let's say this is another image of our particle. If we then calculate its Fourier transform, then these amplitudes and phases also lie upon a central section through the three-dimensional reciprocal space. And because of that, the amplitudes and phases on this plane from our second image will share at least one line of amplitudes and phases with our original image, which cut through reciprocal space in this way. And so if we can computationally detect this line, it's called the common line, where the amplitudes and phases are the same, then we can orient how to put in the Fourier transform of our second image, how we can place that in space, where it belongs in that reciprocal space. We know where it should intersect with the first transform, but what we don't know yet is the angle of how much they should be tilted with respect to each other. We still don't know that angle. However, if we then take our third image, so imagine this is the third image of our particle from some random orientation, and we calculate its Fourier transform, then these amplitudes and phases will lie upon yet another plane within reciprocal space. And if we search for the line of amplitudes and phases that the third transform and the second transform have in common, we might find that they have this line in common. Then we can search for which line does this third transform have with the original first transform. And if we can find that line that they have in common, then we have a fully determined system that should reveal how all three planes should be oriented within the reciprocal space. Now we can take a fourth image. These define a fourth plane that cuts through reciprocal space. And so we again find the common line that it has in common, the fourth with the third transform, and the fourth with the second, and the fourth with the first. And it becomes an overdetermined system, and we generate some kind of least squares fit of the best orientations to maximize the commonality of the intersecting lines between all of these intersecting planes. And we can do that with image after image after image, and not just have 4 images, but maybe hundreds of images defining that system. And once we have used enough slices that we have filled almost all of reciprocal space, here we're still only showing 4 planes, but it could be hundreds that basically fill this space. Then you can interpolate those results onto a regular lattice, and using an inverse Fourier transform in 3D, produce a 3-dimensional reconstruction of the object of interest, here a GRO-EL protein complex. And that's the basic idea of single particle analysis. Now, single particle analysis can be done with objects large and small. So one of the largest objects that it's been done on are icosahedral viruses. This is a picture of a reovirus particle recorded in Hong Zhao's lab at UCLA and published here in this 2010 Cell paper. So each of these is an icosahedral virus. That's one. Here's another icosahedral virus. Here's another icosahedral virus. And these viruses, they look almost spherical. They're actually icosahedral, and they're almost 100 nanometers in diameter. And so from an image like this, using single particle analysis, Hong and his colleagues produced this 3-dimensional reconstruction of the entire virus to 3.3 angstrom resolution. And if we zoom in on one particular place within this very large reconstruction, if we zoom way in and look at individual features, we can see that the map has enough resolution to resolve not only the alpha carbon backbone of each of the peptides chains in there, but also distinct side chains. So here's a tryptophan, here's a phenylalanine, here's a tyrosine side chain. So the EM map was clearly good enough to build an atomic model of the virus. And so in this case, single particle analysis allowed Hong and his colleagues to build up an atomic model of the entire virus. And this is shown impressively in this cutaway view where there's multiple layers in this virus. So here green is the outside layer, and you see the turrets in gold. And then if you cut those away, you see an inner layer with other proteins. And here's atomic models of all of the various proteins that are stitched together to form the entire virus. So whereas the first structures of icosahedral viruses were solved by X-ray crystallography, it has become relatively straightforward to solve icosahedral viral structures by cryo-EM based single particle analysis. and many of these reconstructions have now reached resolution sufficient to build atomic models. Icosahedral viruses are very large particles. Single particle analysis has also been used very effectively on particles that we might call intermediate sized. In fact, the example I'm going to show is that of ribosomes. Now, ribosomes are enormous macromolecular complexes, 2.5 megadaltons or larger in different kingdoms. And here, nevertheless, compared to icosahedral viruses, they're medium-sized. And so in this very recent paper, Brown et al. Report an application of single particle analysis to human mitochondrial ribosomes. So here's the raw cryo-EM picture, and you see each of the ribosomes. For instance, this is one ribosome. You see different ribosomes from different orientations. They have frozen in the ice in different orientations. Each of these is a different ribosome. And by finding their relative angles and averaging together, a high-resolution map was produced of these ribosome particles, at least the large subunit. So here's a view of the outside of the particle, color-coded by resolution obtained in different regions of this reconstruction. And then here's a cutaway view through the center of the particle, showing that also the interior structure was resolved. And here again, the EM map was high enough resolution to resolve side chains of important residues in this map, allowing them to build a full atomic model, including novel proteins whose structures hadn't been solved before, of this incredibly important complex. Now, as an example of one of the smallest particles that single particle analysis has been applied to, I'm going to show the example of a membrane channel, actually the trip channel. And this was published fairly recently by Yifan Cheng's lab at UCSF. And so here is the raw cryo-EM image, and these smaller dark spots are individual trip channels imaged in random orientations within the ice. And so by finding the relative orientations of all those particles, they were able to generate this three-dimensional reconstruction shown with the different subunits colored. And this had, again, a resolution sufficient to resolve important side chains. So here is the center of the pore. It's a channel, and here's isoleucines that define the pore of this channel. And this allowed them to build an atomic model of the membrane protein. So these are three of the most successful example applications of single particle analysis to icosahedral viruses, also very large macromolecular machines like ribosomes, and here even membrane channels at just around 250,000 daltons. And they illustrate a number of the advantages of single particle analysis. The first one is that it doesn't require crystallization. And so you can avoid the time-consuming and luck-requiring step of finding conditions in which your object of interest will crystallize. Also, the samples can be partially inhomogeneous. As we'll talk more about later, because particles are imaged one by one in the ice, if you have a heterogeneous population of some particles with some impurities next to them, or perhaps particles in different conformational states, it can be possible at times for the computer to pick out which of all those particles are the ones of interest and produce reconstructions. Sometimes you can pull out multiple reconstructions from the same data set of each of the different conformational states of your particle, for instance. And so the samples can be partially inhomogeneous. Also, physiological conditions are possible. You can take your purified protein of interest in physiologic buffers and plunge freeze it. And that's in contrast to other structural techniques, for instance, X-ray crystallography or NMR spectroscopy, where your sample has to be in non-physiologic conditions, perhaps in order to induce it to crystallize, or maybe to get it to such concentrations that it will give you good signals in NMR spectroscopy. In cryo-EM-based single particle analysis, you can observe your particle in physiological conditions. The next is that it requires just a small amount of the sample. So if you can purify your particle to say 1 mg per mil, theoretically, you'd only need a couple microliters of that purified concentrated sample to apply to 1 EM grid. And then if the plunge freezing worked well and you stuck that into a microscope, you may only need a 10,000th of the surface area of that grid in order to record enough images, say 10 or 50,000 images of your particle, to produce a reconstruction with enough resolution to build an atomic model. And so, in the end, you need just tens of thousands of your particles in order to produce a high-resolution reconstruction in a well-behaved case. And this obviously is much, much smaller than is required by other structural techniques. Also, it can be rapid. Many of the steps can be automated. For instance, Tim Baker has been working on accelerating the single particle reconstruction of icosahedral viruses to the point where after a grid is prepared of plunge frozen viruses, it can be inserted in the microscope, and then the microscope itself can take the pictures required, and then computers take those pictures, do all the image processing, and can spit out a model within seconds. Now, those aren't the highest resolution models, but nevertheless, because all these steps can be automated, single particle reconstructions in well-behaved cases can be produced very rapidly. Finally, it may even eventually be possible to calculate single particle reconstructions of objects in vivo, because the surrounding material is averaged away when you average the images of many, many particles. And so theoretically, we might even be able to take projection images through cells and pick out particles of interest and solve structures of those particles within the cell. Now obviously this starts to intersect with what is possible by tomography and subtomogram averaging, but nevertheless these are potential advantages of single particle analysis. That all being said, there are important limitations in single particle analysis as well. The fundamental resolution limitation, as it is in all of cryo-EM, is radiation damage, meaning that we simply can't fire enough electrons at each object of interest to get a crisp picture of its details before we've destroyed that particle through radiation damage. So we have to take a large number of low-dose, low signal-to-noise ratio images and then average them. But averaging is limited by the precision of how well each individual image can be aligned. And as we'll talk later, there's lots of alignment parameters that are required. And obviously the more precise they can be estimated, the crisper the average will be. And if there's errors in the image alignment or the determination of the orientation of each particle, this will blur the reconstruction and limit resolution. Next, one way to overcome that blurring is to average larger numbers of particles. And so another limitation in cryo-EM is how many particles can be averaged together. Early single particle reconstructions used thousands of particles. And then as the field developed and it became easier to record more and more images, reconstructions started to emerging with tens of thousands, even hundreds of thousands of particles. And finally, there's a few studies that produce reconstructions with millions of particles that were all averaged together. And the resolution increased as the number of particles were included. But it was also observed that as the number of particles included went up, the resolution reached limits asymptotically. And so you reach a point where averaging more and more particles only very slowly increases your resolution. And most recently, the advent of direct detectors and motion correction has increased the quality of the images included. And so reconstructions with resolution good enough to build atomic model have been obtained with just tens of thousands of images. And so while including just tens of thousands of images is no longer a big challenge in terms of either microscope time, nor computer storage ability or computing time, still the number of particles included in an average is a fundamental limitation of the resolution. Another fundamental limitation is conformational heterogeneity. Many of the largest protein complexes that we'd like to study are by their very natures flexible. They're doing some job moving this to that or creating different kinds of pockets and interacting with different proteins. So they're intrinsically flexible. And if they have a small number of defined states, then it is possible to record images and then separate the images into each of the different states and solve structures of the different conformational states independently. However, if a particle exhibits a continuum of positions, for instance, a continuum of conformations, then it's difficult to separate those and solve reconstructions at different points along the path. And instead, the conformational heterogeneity, while it can be discovered through cryo-EM imaging, it's difficult to overcome and can be a fundamental limitation to the resolution. Finally, it's been observed that some particles have preferred orientations in the ice, or in the context of an experiment using negative stain, particles can settle on the carbon film with a preferred orientation. And when they have a preferred orientation, not all views are obtained of the particle. And so the resolution, the final resolution, can be anisotropic. And that's a fundamental limitation.